Hello, everyone, and welcome to today's webinar, Hemoglobinopathy Testing. What are the key points a lab should consider? My name is Dr. Matthew C. Wagner of Sabia Electrophoresis, and I'll be your moderator for today. Today's educational web seminar is presented by LabRoots and brought to you by Sabia Electrophoresis. To learn more, visit www.sabia.com or sabia-usa.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Kees Harteveld from Leiden University Medical Centre in the Netherlands. Dr. Harteveld, you may now begin your presentation.
Okay, thank you very much, Dr. Wagner, for your nice introduction. I'd like to tell you something about my daily work, so the hemoglobinopathy diagnostics. Hemoglobin is the most important protein for oxygen transport, and the red blood cells, the erythrocytes, are filled with hemoglobin, which is a tetrameric protein, and the majority is hemoglobin A, consisting of 2 alpha globin chains and 2 beta globin chains. They form a tetramer. Besides this almost 97.5% of hemoglobin A, the cells also contain hemoglobin A2, which is alpha 2 delta 2. The delta globin chain is very much like the beta globin chain, but it is coded for by a different gene. These globin chains are synthesized because of the expression of the globin genes. And the globin genes are very much alike. They all consist of 3 exons and 2 introns, and they are located in so-called gene families or gene clusters. The beta globin chain is coded by the beta gene, which is located on the short arm of chromosome 11. And the alpha globin chains are coded for by 2 alpha genes located on the short arm of chromosome 16. And during evolution, the alpha genes have been duplicated. So together, the 2 alpha genes together, they are responsible for the synthesis of the alpha chains in an equal ratio as the beta, the single beta is for the beta globin. So there's a balance between the alpha and beta globin chain synthesis. Diseases which are related to hemoglobin are thalassemia and sickle cell disease, which are, I think, the major ones. So thalassemia is due to a defect of expression of the beta gene or the alpha gene. Now, the beta gene defect gives beta thalassemia major, which is the most common disease phenotype. And this is due to the malfunctioning of 2 beta genes. So the expression is severely reduced, and the alpha globin chains are in excess. They may form tetramers with themselves, which are not soluble, and they precipitate, destroy the membrane, and this causes hemolysis and an ineffective erythropoiesis. And this is the basis of the disease. Sickle cell disease, the other disease related to the beta gene, is due to a missense mutation. And homozygotes for the missense mutation, in which a valin is substituted for, where normally there would be a glutaminyl acid, you get hem S, which is alpha 2 beta S2. And this hemoglobin S forms polymers at low pH or low oxygen tension, and this means that the hemoglobin is actually making polymers. So from solution, it becomes solid, and this distorts the red blood cells, giving the characteristic sickle cell shape. And this also means that the erythrocytes lose their flexibility, so they can cause obstruction of the small vesicles. And this goes with an intense pain, the so-called sickle cell crisis. These disease states of hemoglobinopathy are recessive, which means that you have to inherit both defects from both parents in order to get or develop the disease. Carriers, on the other hand, in which there's only one defective gene, are clinically silent, and they can function normally. Beta thalassemia trait is the condition in which there's only one beta gene or half of the alpha genes maximum not able to synthesize globin chains, and those situation is only recognized by a very mild anemia. The sickle cell trait, it is even less. You have a completely normal hematology, and you have to do either HPLC or capillary electrophoresis to identify a carrier of a hemoglobin S, for instance. Now, why are there so many carriers? Because almost 7% of the world population appears to be carrier of a form of hemoglobinopathy. Well, this has everything to do with this Plasmodium falciparum, which is a parasite living in the guts of the Anopheles, the malaria mosquito. A part of the reproductive cycle is taking place in the guts of the Anopheles, and part is taking place in the blood of a human recipient. So in this case, it appears that carriers of thalassemia or carriers of S have a selective advantage over non-carriers in being able to survive a malaria infection. So, especially at young age, when many children die of malaria, these carriers seem to be protected against death by malaria. However, the consequence of being a carrier is that two carriers can have a child which is a thalassemia major or sickle cell disease. More than 350,000 babies are born each year with sickle cell anemia and thalassemia major due to the fact that there are so many carriers. And the areas indicated in this map in red are the areas where malaria tropica is endemic. However, people tend to migrate in hundreds of years, and in this way also previously non-endemic areas become endemic for hemoglobinopathy as well. So carriers can be found everywhere in the world. The prevention and management of hemoglobinopathy consists of the implementation of basic health interventions. So management of sickle cell disease, for instance, starts with an early diagnosis, then penicillin prophylaxis and vaccination, and this all to reduce the excess mortality amongst children under 5 years. Management of thalassemia major consists also of early diagnosis to start blood transfusions and chelation therapy. Chelation therapy is necessary to prevent the effects of iron accumulation because of the high transfusion regime in which, let's say, milligrams of iron are introduced into the body by each transfusion and don't leave the body that easily. So you need to do something to get rid of that excess of iron. And this is all to prevent unnecessary complications of treatment. The prevention of sickle cell anemia and thalassemia major plays a role in many endemic countries. They do carrier detection, and they provide information and counseling to couples at risk in which both parents are carrier. And this is all to also to offer prenatal diagnosis, of course, if that is legally permitted, and that is not the matter in all states. In some countries, there is a premarital screening and an advice not to marry in case of a carrier situation in both parents. But in the Netherlands, we offer also prenatal diagnosis for this disease, as in many European countries, endemic countries like Italy, Greece, Turkey, etc. And this is to reduce the number of affected births. Hemoglobinopathies is one of the few genetic diseases which can be prevented by screening carriers. Usually, carriers of genetic diseases can only be detected at the molecular level, and screening DNA at DNA level is often very expensive or complicated. However, carriers of hemoglobinopathy are healthy, but they are recognized at the hematological level. And as you probably all know, if you are a little bit, let's say, tired, and you go to your general practitioner, you often get a blood investigation, and the values of that are automatically determined in most laboratories. So every clinical geneticist or clinical chemist can actually see if someone is a carrier of a thalassemia, especially when the iron level is normal. So carriers are easy to recognize at the hematological level, and that is mainly carriers of thalassemia. Carriers of sickle cell disease and hemoglobin C and D, etc., they are recognized at the level of hemoglobin typing, so HPLC or capillary electrophoresis. Carriers can be counseled preconceptionally. That is the best method to do, or the best moment, but it can also be done at the first intake at pregnancy, like is done in the United Kingdom. And a third option is neonatal screening, but of course the baby is already born and sickle cell disease is already found. But if you could also identify carriers in this way, and that is possible technically, and you would report back to the parents that the baby is a carrier, you could prevent an affected birth for a second pregnancy. Many prevention programs, they concentrate on carrier screening and providing genetic information, counseling of couples at risk, offering prenatal diagnosis, and optional prevent birth of an affected child. The hemoglobin switch is an important thing to understand if you want to know what hemoglobin types you can see at the separation level. So if you are using a capillary electrophoresis or HPLC for detecting hemoglobinopathies, it's good to know about the switch. The hemoglobin switch shown in this graph indicates that the genes which are located in order of expression on the short arm of chromosome 16 in the alpha globin gene cluster and on the short arm of chromosome 11 in the beta globin gene cluster is that actually there is a different genes expressed at a different stage of development and also in a different tissue. So first you have the expression of zeta and epsilon in the embryonic phase, and that is, as you can see in the graph, the expression of zeta and epsilon go down rapidly because these genes are switched off, and the alpha gene, on the other hand, is switched on. And for the beta, it's the gamma which are switched on. So during the whole fetal period, you have expression of alpha and gamma, and this forms hemoglobin F. Hemoglobin F is alpha 2, gamma 2. Around birth, you see a steep switching off of the gamma genes and a switching on of the beta genes, which is responsible for, let's say, the synthesis of beta globin, and which forms with alpha globin the hemoglobin A, the adult hemoglobin A. And later on, you see also hemoglobin A2 coming up. This is happening in a period after birth, up to, let's say, 6 months after birth. You have a complete expression of hemoglobin A, and you have hardly any hemoglobin F, remaining hemoglobin F. So this also means that if you have a problem with your beta gene, the disease is going to be expressed 6 months after birth or 1 year after birth, when the hemoglobin is totally dependent on the expression of the beta gene. So at 6 months or 1 year, you can expect the first crisis to occur in case of sickle cell disease, or you get the severe anemic situation in beta thalassemia major. Now, what is the best moment or the best way to detect thalassemia and sickle cell disease carriers? Well, there are 3 components in the lab which play an important role. And that is the red cell hematology, the hemoglobin typing, and the DNA confirmation. And this together will tell you the story if the phenotype fits, or that you have to do more. So for the red cell hematology, we go into detail in the next slide. We measure the hemoglobin content, the red blood cell count, the packed cell volume, which is in my language hematocrit, and the MCV and MCH. For hemoglobin typing, it's important to determine the percentage of hemoglobin A2 to distinguish beta thalassemia carrier, the percentage of hemoglobin X, that could be S or C or D or any of the more than 1,200 hemoglobin variants that we know about, and hemoglobin F, the remaining fetal hemoglobin. And DNA confirmation can be found by using techniques like Sanger sequencing. The genes for alpha and beta are very small, so it's reasonably easy to sequence them completely, including the regulatory elements intons and exons. Gap PCR, which is a way to detect deletions, common or known deletions, namely alpha thalassemia deletions, and MLPA, which is a technique to determine uncommon or rare deletions or duplications. And this reaches a high sensitivity. Most cases of carriership can be solved using these 3 elements. Now, let's first look into the hematology. I have here depicted the hemoglobin percentage, gram percent. In the first column, you see the normal men and women. In the second, you see the carrier. And in the last column, you see the patients with beta thalassemia major. And we are looking, in fact, for red cells which are smaller than usual and more deprived of hemoglobin than usual. So microcytic hyperchromic red cells. We do that by measuring the MCV, which is the mean cellular volume. In the normal situation, it's about between, let's say, 85 and 95. But in carriers, you see it is much lower. So the red cells are smaller than usual. There's a large overlap, however, with patients. If you look at the MCH, that is the graphic below this one, you see that there is also a large distinction between the normal and the carrier of beta thalassemia. And you see that the MCH is considerably lower. So if let's say the red cell is has less hemoglobin, so it is less colored under the microscope, you could say. If we go to the left, you see the red blood cell count, and now we see something remarkable. If you look at the red blood cell count in the normal in comparison to the carrier, you see that the carrier has much more red cells. And this makes that the hemoglobin is in fact, which is above it, the hemoglobin is in fact very comparable with the normal situation. So this means that there is a kind of anticipation of the body where the carrier has, let's say, smaller red cells and more deprived of hemoglobin, but there are many more of them. So this compensates and you have quite a good hemoglobin, which makes that you can do your work. This, however, does not account for the patient. The red blood cells are lower than normal, and that's because of the hemolysis. You lose red cells because of the excess of alpha globin chains. Of course, I could tell you the same story for alpha thalassemia. However, because we have 4 alpha genes, you can either miss 1, 2, 3, or even all 4 of them. So you have more categories. That's why you see, let's say, more columns. And if you look at increasing number of increasing clinical significance, so let's say less alpha gene expression, then you see that the hemoglobin goes down, MCV goes down, MCH goes down, and the red blood cell goes up. The red blood cell count goes up to a certain point where the hemolysis is going to take over, and then you see that the red blood cell counts are going down. And that is, of course, in the patients with hemoglobin H disease. And last, we have, well, not last, the second, we have the HB typing. So we use in our laboratory both HPLC and capillary electrophoresis. And I'll go into detail about that later on in the cases. You can actually distinguish very easily a normal pattern from a pattern with an hemoglobin variant or a beta thalassemia carrier when the hemoglobin A2 level is elevated. DNA analysis or confirmation is done by a system called GAP-PCR. We have several primers to detect the 8 most common alpha thalassemia deletions, and by a simple agarose gel electrophoresis, we can identify the different deletions very easily. If that is negative and we still have a suspicion based on the hematology, we can do sequencing of the complete alpha and complete beta globin genes. And we do the same if we see a hemoglobin variant, because the variant can be either an alpha variant or a beta variant. So we sequence the alpha genes and the beta genes. They're relatively small. So in 4 overlapping fragments, we can sequence the complete gene. And if that does not provide the answer, there might be a deletion at stake. And we detect this with multiplex ligation-dependent probe amplification, abbreviated as MLPA. Here you can see copy number variations, both deletions, but also duplications of certain gene areas. Now, how does capillary electrophoresis and HPLC fit into the diagnosis of hemoglobinopathy? Well, here are some common separations. In the upper part, you see an HPLC, in the lower part, a capillary electrophoresis of the same patient every time. So if we look at the left, the normal gives hemoglobin A and A2, 97.5% about, and 2.5%. If the A2 is elevated, this is typical for beta thalassemia trait. And if you have, for instance, hemoglobin F in an adult, then this is usually due to a deletion taking away delta and beta thalassemia. As you can see, HPLC separates differently from the capillary electrophoresis. So if you have a hemoglobin variant, which you can't see with one technique, you can probably see it with the other one. So they are complementary, let's say. Here are some other examples of common hemoglobin variants worldwide. That is, for instance, carriers of hemoglobin C, and you can see immediately that C separates differently from S and also differently from D, even though S and D, they are, let's say, almost in the same zone, but they are separate. One is in the Z5 zone and the other in the Z6 zone. So they are distinguishable. However, how sure are you if you see a peak in a certain zone? How sure can you be that it is indeed hemoglobin S, for instance? Well, if you have 2 separation methods, that is already, let's say, helping a lot, because you have different separation techniques. One is affinity chromatography, and the other is based on charge. But you have also a confirmation test like sickle cell test. So if the sickle cell test is positive, you know that it is let's say, that's hemoglobin S. And likewise, you can have hemoglobin C, which is different, but then confirmation needs to be done on DNA level. And especially in these cases when separation patterns are much more complex, you need DNA and you need also have some background in genetics. Genetics is fortunately very simple, even though if you look at this graph, you might doubt my words that it is simple, because there are lots of hemoglobin genes and a lot of proteins which can exist. However, we can simplify this. So in adults, you just have the 4 alpha genes to take into account, the 2 beta genes and the 2 delta genes. So what is the maximum of globins you can make from these? Well, actually, hemoglobin A, the most common one, and hemoglobin A2. So these are all the tetramers which you can make from these combinations. And if you have a carrier of S, you will see an extra peak appearing, and that is hemoglobin S, which is about 40% in a normal carrier. Now, alpha thalassemia is one of those thalassemias you can identify easily in a carrier of S without doing DNA analysis almost. So if you look at the normal S carrier, usually the hemoglobin S is 40% about. However, if there is a single alpha gene defect, you see that the S percentage is lower. It lowers up to 35% about. And if you have 2 alpha gene defects, then you, either in cis or in trans, you see that the hemoglobin S goes down to about 25 to 30%. And that is very well distinguishable with capillary electrophoresis. The HPLC, you can also see it, but it is because of the let's say, the baseline, which is less stable. HPLC, it's more difficult to see it, but capillary electrophoresis is it's very well visible. You can almost put a line to see that there is actually also alpha thalassemia playing a role. Okay, I'll go through the cases now. I have 7 cases. One case is the genotype-phenotype correlation in this African male. He has microcytic hyperchromic anemia. We see a lot of peaks, and DNA analysis shows us that this patient is homozygous for the so-called 3.7 deletion, a rightward deletion, which means that he is missing 1 alpha gene on each allele. And on top of it, he has a point mutation in one of those, causing G. Philadelphia. That is let's say the protein which is due to a substitution of amino acids. So G. Philadelphia it's called. And he is S carrier, which is probably not a surprise because of his African origin. So if we look at this picture and determine how many different hemoglobin types you can predict from this composition of DNA, well, at least the normal ones, so the hemoglobin A from alpha and beta, the hemoglobin S, alpha to beta S2, and alpha to delta S2, but of course also the G. Philadelphia can form these combinations with beta, beta S, and delta. So in total you have 6 different hemoglobin types, and all of them you can trace back on the pattern you see on the left side. So in this way, it is you can see that all the hemoglobin types are there, and you can easily understand. Well, sometimes you have bad luck, and you may miss something with your HPLC. Like in this case, you see on the right the HPLC pattern, and we see actually a completely normal pattern. But on the left, using capillary electrophoresis, we see a small peak just in front of the A. And this is due to an alpha variant, this alpha variant of alpha 1. So in principle, you can expect the normal hemoglobin A and hemoglobin A2 but also the alpha variant of the, let's say, alpha beta and the alpha delta. So there's also an extra A2. And in fact, if you look carefully, you can see here the A2 peak, but there's also a very small peak here, which added up is giving the total hemoglobin A2. This is hemoglobin NUAC shot. It is visible on CE, but not on HPLC, and it is an alpha 1 variant. On hemoglobin VAR, it indicates that the website where the hemoglobin variants are presented, it is 25%, it's unusually hydrophobic, and no clinical consequences are related to it. Well, we have seen 2 Dutch families with this hemoglobin NUAC shot, and that gives about 9% of hemoglobin X. The second case here is a male with a slightly microcytic hyperchromic anemia, and we see, let's say, a distinctive peak between the A and the A2 on the capillary electrophoresis, but the HPLC shows a little bit strange peak here. It is actually under the A. It's hemoglobin Atlanta. It's visible on capillary electrophoresis, but not really on HPLC. So it would be missed easily on HPLC. And this is an unstable hemoglobin variant, giving mild hemolytic anemia in the carrier. But it could also be the other way around, unfortunately. So something like this, you can see a completely normal capillary electrophoresis pattern, and now the HPLC shows something strange. And this is the hemoglobin Santa Guana. It's a very rare one, but it is a hemoglobin variant of the beta chain, and it gives about 10%, but this is a bad estimation because of the high peak underneath, and this is due to the overlap. It's a hemoglobin variant in which the oxygen affinity is decreased, it gives mild anemia, and is found mainly in Mexicans and in Greek. For a hemoglobinopathy reference lab, it is therefore convenient to have both capillary electrophoresis and HPLC. However, all common clinically relevant hemoglobin variants, like hemoglobin S, C, D, E, etc., separate very well on capillary electrophoresis or on HPLC, so you don't need both necessarily. Well, this is an example in which we see a double peak in the capillary electrophoresis, so there is a split peak here, and HPLC shows it in a better separation, and this is due to a beta variant. And this beta variant is hemoglobin Hounslow. The codon 80 is changed, and you see a tyrosine instead of an asparagine. And this is present in about 38% in the heterozygote. It originates from Afghanistan, and it gives a normal clinical presentation. So in this case, there's no clinical consequences and also no genetic consequence. Hemoglobin variants sometimes travel around the world. And I would like to show you an example of this one. This is hemoglobin Malmö. Hemoglobin Malmö is a beta variant in which a alpha-1 beta-2 contact is disrupted because of the substitution of a histidine for a glutamic acid, glutamine. Sorry. This goes with the erythrocytosis. So and we also have a Dutch family. However, it was firstly described in a Swedish family. And probably you recognize these fellows. These are Vikings, and they come from Sweden. And in the year 1000 and 900s, they raided the coastal areas of Europe and left their genes there. And they burned everything to the ground and dragged home all valuables. So this hemoglobin from with the increased oxygen affinity causing polycythemia was maybe of some benefit to these gentlemen, because they were, let's say, more highly and They could, let's say, bind their oxygen very, very well, and maybe they had the possibility to go on forever and ever. However, this hemoglobin Malmö has also a disadvantage, and that is that carriers often experience, let's say, headaches or and bleeding of this is helping just to reduce the PET cell volume. Now, hemoglobin Malmö was first described in the Swedish family, but it was also found in the Netherlands, also in England from a Saxon family, and from a French family coming from Brittany. And last but not least, in Sicily. And what many people don't know is that the Vikings actually reached Sicily and had a kingdom there for over 200 years. So in this way, you can almost trace back the root of the Vikings by looking at the hemoglobin. Sometimes the hemoglobin variant is very unstable and therefore not detectable at a protein level. This causes a beta thalassemia trait phenotype. As it happens in the hemoglobin or in the beta gene, you'll notice an elevated hemoglobin A2. Like in the case of hemoglobin Showa Yakushi. That sounds very Japanese, and it was also firstly described in a Japanese female. Also in a Japanese family, there was a proband mother and sister and an unrelated Japanese individual. Later, hemoglobin Showa Yakushi was also found in some families from West India. But we found it in a Dutch family originating from a small village near the Dutch coast. And many coastal villages, there were sailors. And sailors, they were going around the world, let's say, and especially in the Dutch East Indian Company time, into 16th and 17th century, there were a lot of settlements of the Dutch East Indian Company in the different countries where trading was happening. Also in Japan. Here are some characteristics. It gives hemolytic anemia in the carrier, and the elevated A2 is very typical. So it looks like a beta thalassemia trait phenotype, but actually it's an unstable hemoglobin. And it gives, let's say, beta plus thalassemia. Now, the question which arose was, is this hemoglobin Showa Yakushi, which happens to be Dutch, but also in the Japanese and the West Bengali, have they some association with each other? Are they the founder, maybe? But how did it come about? So could it be a rare Dutch hemoglobin, maybe, variant traveling to Japan or West India, or the other way around? And for this question, you need to determine the haplotype. And the haplotype of the Japanese hemoglobin Showa Yakushi mutation was determined. Now, I have to tell you that the Dutch were actually traders, and Japan was in the 16th, 17th century very closed. The emperor did not allow strangers on Japanese ground. So he had an island constructed, an artificial island in front of the coast, and the Dutch were supposed to stay there. And just to prevent them from escaping, the emperor sent to this settlements. Geishas were Japanese ladies, and marriage came about between the people from the company and the geisha, and also children were born. There are, let's say, there's historical information for that. However, when the employee had to return home to the Netherlands, he was not allowed to take his wife and children with him. So that means that the, let's say, gene flow was actually from Holland to Japan and not the other way around, in theory. So we investigated the haplotype of the West Indian, the West Indian sample was already investigated, and the haplotype was published, and it was different from the Dutch and the Japanese haplotype. However, the Dutch and the Japanese haplotypes were identical. But this identical haplotype was the most common. So there's still a large chance that it was purely coincidental that the mutation occurred twice on independent genetic backgrounds. But a founder effect might also be possible. We don't know, historical proof is lacking. So we don't know whether this ought to be a hemoglobin VOC from the Dutch Indian Company abbreviation, but I think we should stick at the Showa Yakushi. Talking about elevated A2, there are a lot of stories going on about the importance of hemoglobin A2. We know it's from the beta thalassemia carrier, but also many people ask, if beta thalassemia trait goes elevated A2, does that mean that alpha thalassemia trait goes with a decreased hemoglobin A2? Well, from this picture, you can see that actually iron is lowering the A2, iron depletion. However, the alpha is not really affected. The A2 is not really affected that much. There's still a large overlap with the normal controls when you lack the expression of 1 or 2 alpha genes. If you lack the expression of 3 alpha genes, then it is reduced, but then you also see hemoglobin H. So I don't see really much added value to the hemoglobin A2 in the relationship of diagnosing alpha thalassemia. Of course, without doubt, elevated A2 goes with the beta thalassemia trait, even in the presence of alpha thalassemia or in the presence of alpha triplication. That does not really influence the A2 very much. On the other hand, if you have a beta thalassemia in combination with delta thalassemia, then the A2 goes back to normal, and then you will probably consider this as a microcytic hyperchromic anemia with normal A2 representing an alpha thalassemia, but in fact it's a combination of delta and beta thalassemia. There are also some beta variants which go with a almost normal A2. We'll come back to that here. You see here a group of, let's say, normals in red. This gives a normal A2 and elevated A2. These are beta thalassemia trait confirmed by DNA analysis. However, you also see a kind of overlap. And if we focus on the overlap, it appears that these are all elevated or almost A2 situations of a particular type of beta variants, of beta thalassemia mutation. They are listed here. So the IVS1 6 D2C, the +1, -101, etc. These are all mutations in which given heterozygote, only a very tiny elevated A2. So they can easily be overlooked. So an elevated A2 can also be inconclusive. In this case, case 6, is that the beta thalassemia trait seen in these persons in this family have an elevated A2, a microcytic hyperchromic anemia. However, there is no mutation found in the beta gene. So we have looked at the alpha-beta chain synthesis ratio, and that was, let's say, disturbed in one of the family members. So it is a true beta thalassemia according to the definition, but there is no mutation in the beta gene. Moreover, from whole exome sequencing, we also found that there was no correlation with the beta globin gene cluster. There was, however, a correlation with a gene called SUBT5H. And this SUBT5H is showing a mutation in the splice donor site and in the other family, the splice acceptor site, which gives on RNA an partial intron retention, and therefore a disruption of the reading frame. So the protein is in fact shortened in the most important region. Here you can see the C-terminus of this protein. The SPUT5 protein is the product of SUBT5H, and that is let's say, not made in these carriers. And this protein plays a part in the DCIF complex. And this DCIF complex is both activator and repressor of transcription elongation. So if the phosphorylation cannot take place because of the absence of this repetitive area on the C-terminus, you get that it is blocked. And this might be the reason why these people have an elevated A2 and, let's say, a typical beta thalassemia trait. We don't know for sure, but animal studies, like in zebrafish, indicate that this lumen SPIT5 is active in the hematopoietic erythropoietic specific region and tissue, and that it is also playing a role in GATA1 induction. And from GATA1, it is known that it plays a very important role in the regulation of the beta gene. In the meantime, we have found 8 different SUB5H mutations in 25 persons, all showing this unlinked beta thalassemia trait phenotype. And all of these 8 mutations are null alleles, so they just do not allow the expression because of frame shift mutations or stop mutations. Last case is one which we obtained from neonatal screening. You can see the switch here. This is around birth. The normal situation, you have in a baby the active alpha genes, the beta genes, and the delta is not active yet. And we have the gamma, which is expressed and gives hemoglobin F. So around birth, a normal baby is about 80% hemoglobin F and some 20% hemoglobin A. Now, if you have a single alpha gene defect, we see that there is a relatively excess of gamma, which cannot find an alpha partner, let's say. So they start forming gamma tetramers with self-hemoglobin baths in a very low percentage. But in, let's say, the neonatal screening, that is a good marker for the presence of alpha thalassemia. I tell you this to understand better the following case. We found this baby in the neonatal screening in collaboration with Ampia Hospital in Breda in the Netherlands, and this is a strange pattern. This baby was 18 days old when it was presented, and we did DNA analysis of this one, and there appear to be 3 defects. One is the 4.2 deletion, which is a so-called leftward deletion. It takes away 1 alpha gene, and on top of it there was the hemoglobin Q-tilent mutation. So on that allele there was also a missense mutation. So this gives a chain, the so-called Q-tilent, alpha Q-tilent chain from this allele. And the baby was carrier of hemoglobin E, which is very common in Thailand and Cambodia and Vietnam, etc., so in East Asia. So if we look at this composition and estimate how many different hemoglobins, tetramers, can be formed with this genetic composition, you can see that you have the normal alpha with the beta, the normal alpha with the beta E, the alpha Q-tilents with the beta E and with the normal beta, and of course you have your gamma still, because that is still expressed after birth. So your normal hemoglobin F and your Q-tilent hemoglobin F. And besides that, you also have the hemoglobin bars. Okay. And do we find this back? Well, in fact, you see 6 different peaks, and there are 7 different tetramers. And the reason is that the FQ-tilent is actually, let's say, the normal hemoglobin F coincides with the Q-tilent hemoglobin. So they're on the same position. So they go together, and that's why the so-called F peak is not really F. It is F and Q-tilents. So we look at the next slide. This is a situation of the same baby, but then at 87 days after birth. And you see a changing of the pattern. This is, of course, visualizing the switch, because in the meantime, the delta is come up, the gamma expression goes down, and the beta is going up. So now you have not anymore the hemoglobin bars, because that disappears in such a mild alpha thalassemia, but you have the appearance of hemoglobin A2 and the hemoglobin Q-tilent A2. So now you have here the different peaks. There are 7 peaks and 8 different hemoglobin tetramers, again because 2 are overlapping. If we look at the pedigree, you see the boy, the neonate, at 2.5 weeks and 3 months, and you see his little sister, who's 2 years old. And if you look at the pattern of the sister, and you look at the pattern of the father and the mother, it's almost as if the sister has the pattern of mother and father superimposed. And this is also what happens to the boy. After, again, 3 months, he will look exactly like his sister. And the good news is that this is not a bad situation. The combination of having a hemoglobin E trait with Q-tilent doesn't really matter. Q-tilent is a neutral or silent hemoglobin variant, and the fact that the boy has an alpha thalassemia and a hemoglobin E, which is a hemoglobin variant with a beta thalassemia trait effect, actually the balance becomes again quite good. So this means that there is no clinics for this child. In conclusion, what are the key points a lab should consider in hemoglobinopathy diagnosis? Well, capillary electrophoresis and HPLC or HPLC is not enough. It is a combination of the red cell hematology, the hemoglobin separation, and the DNA confirmation that makes the diagnosis complete. An elevated hemoglobin A2 is a very good indicator for beta thalassemia traits, but DNA confirmation is still important, as not all elevated hemoglobin A2 cases apparently carry a mutation in the beta gene, and the only way to find out is sequencing the beta gene. Common hemoglobin variants can be detected easily both by capillary electrophoresis and HPLC, but can only be characterized at the DNA level. And then, of course, perhaps the exception is hemoglobin S. Hemoglobin S can also be confirmed by the sickle cell test. A positive sickle cell test is a good indication that it is really S. Also, hemoglobin E, the DSIP test is done for that. In Asia, it's very frequently done. It gives a turbidity when the hemoglobin E is present. But alternative tests are, in my opinion, not available for hemoglobin variants. So it is wise to do DNA confirmation in those cases. So this means that you have hemocytometry, you have capillary electrophoresis, and it can be CBR, of course, DNA, and that should fit the last puzzle piece of the clinical diagnosis. If that doesn't fit, there's something you overlooked, and you have to repeat the analysis or look for special things like copy number variations and the alpha genes, for instance. I would like to acknowledge all my collaborators in the Netherlands and abroad. Thank you very much.
Hey, thank you, Dr. Harteveld, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now.
Hey, thank you, Dr. Harteveld, for that informative presentation.
Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. I see we already have a few questions. I'll just go through them in order here, Dr. Harteveld. Could you give some examples about hemoglobinopathy screening? This may have been answered. This is a question from Bassem Sharia from Yale University. And I believe what they're asking for is just the different methodologies of hemoglobinopathy screening. Dr. Harteveld?
Yes, yes. Well, there are indeed different forms of screening. In the Netherlands, we do neonatal screening, and that is HPLC screening. So the newborn babies, the blood is collected in Guthrie cards, and then it is dissolved, and it is tested with HPLC. In this way, it is possible to see carriers of hemoglobin S. These are also reported, and of course, babies with sickle cell disease. And the screening is mainly to see disease phenotypes, so thalassemia major and sickle cell disease. However, as carriers are easily detected, these are reported to the parents. But it was not the intention of the screening program. The intention was just to focus on sickle cell disease and thalassemia major. Other ways of screening carriers are not organized in the Netherlands. That means that it is just the idea of the midwives, for instance, to collaborate to see if they want to discuss this with their patients. So in this way, it is possible, for instance, if there's a hemoglobin checkup, which is standard amongst pregnant women, you can also ask for an HPLC or a capillary electrophoresis analysis at a local lab. And in this way, carriers can be detected. And we try to promote that this will be discussed, at least with the pregnant and the husband.
Mm-hmm. Okay. Moving on to the next question. We have a question from Michael Cicero and Maimonides. I'm probably saying that wrong, Medical Center. As a teaching hospital, can you remark on H bodies seen when red blood cells are stained overnight with a supravital stain when R/O hemoglobin H trait or disease? I'm afraid I don't know the R/O abbreviation, what that stands for.
Yeah. So the question is if you can see hemoglobin H inclusions with the brilliant crystal blue staining. Well, we do that. We do perform this test, but usually it is only to support a DNA analysis. So in the case we see a, let's say, clear microcytic hyperchromic anemia with a normal A2, and we don't find anything on DNA level, and still we have a proper suspicion that it must be alpha thalassemia, then hemoglobin H inclusions can help to make sure that you have to do, for instance, MLPA analysis, because then you are dealing not with a common alpha thalassemia, but with a deletion type or a rare deletion type. So we use the hemoglobin H inclusion bodies test, but mainly to support our DNA analysis.
Okay, thank you. Question from Terence Agbor from Dynalife Diagnostic. How does hemoglobin Nuakchot run on acid and alkaline gel electrophoresis? Do you happen to know?
No, I'm afraid I don't know that. But I think it's an interesting question because these rare hemoglobin variants, well, they can be, of course, sometimes missed, and you would really like to have more systems in your hospital or in your laboratory. However, we are already quite blessed with the fact that we have an HPLC and a capillary electrophoresis. So I can't ask for more from my superior, I'm afraid.
In my own just quick perusal, I happen to know that Nuakchot migrates with F on isoelectric focusing, which is roughly analogous to its position on alkaline, but I'm afraid I don't know where it migrates on acid. Another question from Michael Cicero at Maimonides. We use cellulose acetate and citrate agar to identify our hemoglobinopathies to date. To confirm, for example, hemoglobin DG co-migrators under S on cellulose acetate, we submit for alpha chain analysis to confirm hemoglobin G until we are approved to upgrade to HPLC. What is your opinion of our use of the current method?
Well, I have no experience with the citrate methods.
Mm-hmm.
So I think what is the most important thing is that you're able to identify, let's say, the most common ones. And if you have, let's say, good experience with the citrate in combination with other methods, I think it is just a very good method because it indicates that something is there. However, the typing of the hemoglobin variant is still debatable. I mean, there are so many variants, and there's a chance that even if you use two methods, you may not be able to identify which hemoglobin variant it really is. So in this case, I would recommend DNA analysis as a following step.
Okay. Another question from the same asker. Adult pre-op India showed hemoglobin E disease. Siblings the same after study. Prognosis? So I think this is asking for prognosis of hemoglobin E homozygous disease.
Well, with hemoglobin E homozygotes, usually these are clinically mild, so no treatment is necessary. However, if a pattern shows hemoglobin E homozygosity in a patient which is transfusion dependent, you may have a beta thalassemia on the other part. So there is a possibility that you have an E beta 0 tau. In that case, the clinic is much more severe, and these patients present with beta thalassemia intermedia, so they may occasionally or regularly need blood transfusions. At a molecular level, they are easily to distinguish because you usually have, 95% of beta thalassemia as a point mutation. So if you do the sequencing analysis, you will see heterozygosity for the E mutation and a heterozygosity for an additional beta thalassemia mutation, so a compound heterozygote. But in case you see a homozygous pattern, I would really recommend to do MLPA analysis in those cases, because then you might have a combination of a beta E allele and an beta 0 thalassemia deletion type.
Okay, thank you for that. Two related questions from Theodore Kurtz at UCSF School of Medicine. What online resources do you recommend for databases showing where variants migrate on capillary and on HPLC?
Well, that is also a very good question. I know of databases showing DNA results, and hemoglobin VAR sometimes also gives some information about the different separation methods at protein level. But what I think might be useful is the atlas that CBI is providing to its clients. There you can find a lot of different patterns in relationship to the DNA genotype. So I think that would be a way to go. But I know that Ethernet, that is the database which is curated by let's say a collaborating group of experts. That is also quite good up to date, but it does not really provide you requested. So HPLC patterns and capillary electrophoresis would be very, very nice if that would be possible.
Mm-hmm. I should note we are at the top of the hour here. I think we'll be able to answer a few more. There's a question here which I have encountered hemoglobin, I believe they meant to say hemoglobin F in infants past 6 months of age. What is the follow-up, and what kind of comments may be included in the hemoglobin electrophoresis screening report?
So if I understand well, it is an infant of 6 months old and is expressing just hemoglobin F.
It's a little unclear from the question. I believe they actually said they have encountered hemoglobin A in infants past 6 months of age, which of course would be would be the normal state. I believe they meant to say hemoglobin F, so a high persistence fetal hemoglobin or something like that.
All right.
Right.
All right. Well, if you find, let's say, a high percentage of hemoglobin F, it would always be good to check the parents, because it is an inherited condition. So one of the parents should have showed the same, or if it is, for instance, due to a beta thalassemia defect in a homozygosity or compound heterozygosity, you should see it in the parents as well. But if it is hereditary persistence of fetal hemoglobin or HbFH, one of the parents should be carrier at least. So I think it is important to do either DNA confirmation or DNA diagnosis on these cases using MLPA for the beta cluster to determine HbFH deletions, or if their deletions are not present, the gamma promoter sequences, just to detect the non-deletion type of HbFH, which are in fact quite rare. Delta beta thalassemia deletions are much more common in this respect. Everything is relative, of course. But if you think that the hemoglobin F is due to a, let's say, disease-related mechanism, so not HbFH, but actually disease-related, so just F expression, for instance, in a small child, it's very likely that it is beta thalassemia major, and I think in that case, screening of the parents would be very helpful.
A question about a particularly sticky issue from Claudine Bates, Central Laboratories, Bailey Road. Can you please explain the new drug voxelotor, why this drug interferes with our capillary hemoglobin assay?
Well, I have no experience with that, so I can't speculate on that, I'm afraid. Sorry.
Okay. Any advice for diagnosing beta thal trait in newborns? This is from Maria.
That's a very good point.
Harry Medical College.
That's a very good point. At birth, the A2 is not visible, so for a newborn, it is not possible to look for elevated A2. If you would like to have a screening like that, it is either on DNA level, but then you lack actually the information of the HPLC, because that is not really, or capillary electrophoresis, because that is not showing anything. Another possibility would be to screen children at the age of 1 year or older. And this way you could see the elevated A2, of course, easily. And if you don't want to screen children, because, well, at that age, maybe it is not relevant yet to detect carriers, then you could decide to screen children, or let's say adolescents at school age. And that is something which is done in Italy. There's, of course, a high incidence of beta thalassemia, and already in the 60s, school children were screened with permission of the parents to see if they have elevated A2 and are therefore carrier of a beta thalassemia. So that, let's say, advice could be given during biology lessons.
Question from Gillian Pena from NSMBB, the CDC. When using capillary or HPLC, were samples screened or tested in liquid blood, or instead were dried blood spots used?
Well, we use only liquid blood, but the neonatal screening in the Netherlands is all dried blood spots.
Question from Anupam Basu. If alpha and beta, I believe, variants co-associate, what will be the clinical outcome?
Well, actually, if the alpha and beta mutations cause, let's say, an expression defect, so alpha thalassemia, reduced expression of alpha, and the beta mutation and decreased expression of the beta, then actually you restore the balance. So if the if you have a double carrier of an alpha thalassemia and a beta thalassemia, what you see is a higher red blood cell count and a somewhat lower MCV and MCH, but generally the hemoglobin level is quite okay. It's, let's say, at the border of low hemoglobin. So it's not a pathological condition.
Okay. From Terence Agbor at Dynalife, a few months ago there was a CAP challenge consisting of high affinity hemoglobins, Heathrow, Malmo, and Olympia, etc. It's difficult to distinguish these variants by HPLC and capillary electrophoresis. Do you happen to know how these variants migrate in gel electrophoresis, acid alkaline? We run our hemoglobinopathies with CE, then reflex to gel electrophoresis.
I see. Well, I have little experience with the electrophoresis as you do that. So I can only compare the HPLC with the capillary electrophoresis. And yes, sometimes it happens that you can't see an HB variant with one method, but if you see it with the other, it's really no problem as long as you do DNA confirmation.
Yes. Yes, it should also likely be noted the high affinity hemoglobins won't necessarily behave the same way in capillary or HPLC with one another. So Heathrow, Malmo, and Olympia may migrate very differently.
Yes.
Does your laboratory keep inventory of different variants? What kinds of samples? Does the lab perform stability on the samples over time? This is a question from Gillian Pena at CDC.
Well, we don't do really tests on stability of hemoglobins. If we find a hemoglobin variant, sometimes we search the collaboration with another lab who is let's say more specialized in studying proteins, because I'm working in a genetic department. So the fact that I'm looking at protein levels is quite exceptional for my colleagues who look upon me as I'm coming from Mars or something. So if we really want to have more knowledge about the hemoglobin protein and about its stability, then we have to look outside our department. And the collaboration with labs in Paris, for instance, from Henri Wijkman in the past has been very, very useful.
Okay. Question from Patrick Trelette from TOSO. What about the significance and accuracy of the A2 value in case of hemoglobin S or variant in the HPLC technique and capillary technique? Is it correct to report this A2 value?
Well, hemoglobin A2 values are, of course, important to distinguish the beta thalassemia carrier from a non-carrier, but one should be cautious because, as I have shown in the graph between the group of normal A2 and the group of elevated A2, usually there's quite a distinction between normal A2 and elevated A2, but there are also certain mutations which only elevate the A2 marginally. So this means that there is a certain overlap between the normal distribution of normal A2 and elevated A2. So I think it's important to measure the A2 as accurate as possible, but you can never rely on one method. Okay. Whether it's HPLC or capillary electrophoresis, I understand that the screening lab needs a range and a cutoff. But of course, a screening lab takes into account that it will possibly misdiagnose a certain percentage of cases, simply because it is, let's say, one of those beta thalassemia mutations which go with a hardly elevated A2. And if that is very common in the population, they really should be very careful what cutoff range is important. So I think it is important to have an accurate A2, And of course, that is the reason why the world is working hard to have a standardization for hemoglobin A2. But on the other hand, we have to take into account that you can never rely on a single method.
Mm-hmm. All right, and we have one last question to close out, I believe, from Gillian Pena, CDC. Will you be able to share the slide deck? I don't know if you had planned on making your slide deck available beyond just the recording of this presentation.
I think my slides are available, and the recording as well, as I understood from CBI. I have no problem with that.
Okay. I believe that is the last of the questions we have. Thank you very much for your time and for coming back to answer these questions after the recorded presentation. I believe we'll close out now. Thank you very much, Dr. Harteveld.
Okay, thank you very much.